Thanks for joining us today. I'm Macy Heel, and I'm joined by my colleague Aidan Sullivan.
Hi, everyone.
We're here today to share with you how you can use Neurolucida 360 to annotate your microscopy image data with fair, curated vocabularies.
Happy Taco Tuesday. Today we're going to talk about what makes digital reconstructions produced with MBF Bioscience's new vocabulary services fair or bindable, accessible, interoperable, and reusable. We'll show how to access these vocabulary term lists from an API connection to SciCrunch directly within Neurolucida 360, and how to transform previously created segmentations files into fair data. The features that we're discussing today were developed through MBF Bioscience's participation in the NIH Common Fund SPARC, or Stimulating Peripheral Activity to Relieve Conditions. I do see a lot of SPARC researchers here today, and it's great to see those familiar names. We're glad you could join us. But I do want to reassure those of you who are not familiar with SPARC that the tools we're demonstrating today can be used for those researchers too. The functionality in MBF Bioscience software will soon be rolled out to the broader scientific community. The SPARC program aims to accelerate the development of therapeutic devices that modulate electrical activity in nerves to improve organ function. The program has built a web portal, sparc.science, that hosts open data generated and curated by SPARC to encourage the reuse and further accelerate scientific discovery.
The heterogeneous and dynamic data produced by SPARC investigators comes from many organs and species. SPARC investigators that are mapping the autonomic nervous system have access to MBF Bioscience software, such as Neurolucida 360, which we'll use in our demonstrations later, to utilize in their image segmentation and analysis pipelines. The FAIR segmentation data created in our software can be ingested into these 2D and 3D maps seen here on this slide that are on the SPARC portal. We hosted a webinar series last month that detailed the image segmentation to scaffold pipeline. In part 1, Aidan and I demonstrated how to use FAIR segmentation tools in our software to create and prepare files for scaffold integration. If you'd like to learn more, we encourage you to please visit the sparc.science YouTube channel, and you can check out part 1 and part 2. But today we're going to take a closer look at how easy it is to access anatomical vocabularies from within our software and use MBF segmentation and editing tools to add metadata to existing files.
So aside from segmentation for the purpose of fitting to organ scaffolds and maps, users can also derive quantitative conclusions about cellular features and connectivity within their image data. These downstream analytics can be performed in MBF software like Neurolucida Explorer or in custom analysis tools. One of the SPARC project's initiatives is creating open data that can be repurposed and remixed. We want to make sure that these microscopy segmentations have descriptive file-level metadata. This metadata provides a sufficient explanation of what the data is and where it came from and how it was created. Through the novel integration in MBF Bioscience software that we're going to talk about today, you can add this metadata to segmentation files at the time of data creation or to existing files. By enriching this metadata, we can contextualize the data and make it findable, accessible, interoperable, and reusable, or FAIR. The mechanism that we've created works under the hood, so the data you generate in Neurolucida 360 has the necessary FAIR aspects with little effort on your part. These FAIR aspects include metadata about your sample and the structure models that you've created with Neurolucida 360's toolset. The metadata allows the morphological models to be interpreted at the file level independent of you.
Making segmentation data fair is possible through our API connection to the SciCrunch platform. Once connected to SciCrunch, users can access organ and species-specific anatomical vocabularies from curated ontologies to utilize during annotation. By connecting to SciCrunch at the beginning of each tracing session, you can surface the most up-to-date term lists. That means if you've requested new terms to be added to the ontology, you'll see them as soon as they're added without needing to update or install new software or going back and forth from ontology websites like Uberon and FMA. All the terms from SciCrunch have persistent and unique identifiers that are mapped back to those ontologies. These identifiers are written to the MBF data file at the time they are used, so you don't have to do any of the heavy lifting, and I'm going to show you how that works from the user perspective in just a little bit.
To demonstrate employing this terminology in an experimental workflow, we're going to use one of the samples from Dr. John Tompkins' lab at UCLA. This data uses a wide variety of segmentation elements, making it a great demonstration for naming these elements with FAIR terminologies. Segmentation data with FAIR terminologies can be applied to a variety of microscopy image data, though. This particular Spark research lab has used MBF bioscience software to perform multi-resolution image segmentations to understand the structure-function relationships of the cells within the stellate ganglion. On the screen here, you'll see a 40x image of backfilled cells from the stellate ganglion taken with an epifluorescent microscope. Electrophysiological recordings were obtained from these same cells. You can see that the neuronal pathways at the 40x magnification can be easily distinguished and traced. The cell bodies and neuronal fibers were segmented using Neurolucida 360's semi-automatic tracing mode. You can see the complete high-resolution tracing in the video. Dr. Tompkins' lab also imaged the whole stellate ganglion in 3D at 10x magnification. And merge the backfilled cells reconstruction with the low resolution stellate image volume, moving the data in X, Y, and Z until it's aligned and registered to the lower resolution image. The registration process provides context with the larger structure of the stellate ganglion here. Next, the ganglion from the low resolution image was contoured in 2D in a series of planes at a set interval. And you can see the 3D results of that process here. And these contours can be shelled into a 3D surface that represents the whole volume of the stellate ganglia. Additional structures, such as the cardiac nerve, were segmented to provide fiducial landmarks. Next slide. There we go. In this example of a segmentation that allows morphological and physiological data to be registered to a generic organ scaffold using the detailed fiducial markings. However, it is not complete until these fiducial structures segmented in the stellate ganglion are named using FAIR ontologies from the SciCrunch database. With these FAIR terminologies embedded into the data file, it is easy for researchers to communicate the relevant anatomies to the scaffold engineers and ensure appropriate registration of the segmentations to the 3D structure models. These anatomies can also be surfaced on the Spark portal using the scaffold viewer to help communicate the specific terms to the portal users. So let's dive into how the Tompkins lab makes the data files like this one FAIR using Neurolucida 360's connection to the SciCrunch terminology database. Take it away, Macy.
Thanks, Aiden. So here you'll see I have the Neurolucida 360 program window open, and I'm going to use the data file that Aiden just showed in that last video to demonstrate how you can take existing data, or maybe data that's been converted from another segmentation source into MBF XML, and add these FAIR annotations. So using SciCrunch and the vocabulary terms to make each individual element more fair. So to start, we'll bring in an XML file, and the XML file is associated with the image data. So we'll drag and drop. I apologize, the screen got a little small there. There we go. And when you open a data file, you'll see this welcome window appear. It's split into two sections. One is the subject information. That's where we're going to write subject-specific metadata to your file. We'll indicate the species, subject ID, and the sex and age of the sample. You can see here that the fields have already been auto-populated. This is because we've already created this data file previously in Neurolucida 360, so it's going to remember this information. There is an option for blinded to condition for sex and age. If you are blinded to the experimental conditions at the time of annotation, you can go back in and change that to the actual sex and age later on and save that, and that metadata will override the existing metadata and save to the file. On the right-hand side, this is where we're going to communicate with SciCrunch. So we're going to indicate the criteria for the anatomic terms we want to retrieve from the database. The first is to select your organ. Here we're using the peripheral nervous system for the stellate, but you can see there are other organ lists to choose from, brain, colon, heart, etc. And then after we select our organ, we can indicate the species, and if a parcellation for that species and organ exists, say the Allen mouse brain atlas, you can choose that within the parcellation dropdown. If no atlas or parcellation exists for that organ and species, you'll see it's species independent, and you'll get a list of all the peripheral nervous system terms for multiple species. We'll hit connect. We're going to now see in the 3D window the 3D image data of the whole stellate with the tracing data as well. Aidan explained how they use contours to contour the body of the stellate and how they trace these high-resolution cells in Neurolucida 360 and then merge them onto the 10x image data. They also traced the nerve bundles that enter and exit the stellate with the tree algorithms. And what I want to show you today is how the SciCrunch terms appear in the software. And we're excited to tell you that they appear in multiple places. So each individual element can be named with a SciCrunch term specific to that anatomy. In the contour selection toolbar here, you'll see I can search for a specific term, in this case, the cervicothoracic ganglion, and I can use that for contouring. I can also use markers. I can rename a marker if I want to say a distinct point or a cell is a specific vocabulary term, and we have a search bar there as well. But what's new in Neurolucida 360 is the ability to use these terms for naming segments or whole branches of trees or fibers. And to do that, we're going to work in the 3D environment window. And we will first go and click on the tree icon on the right-hand side of the program window. I'll show you the segment there that we want to classify these that branch there, the inferior cardiac nerve. And when I hit tree, you'll see there are different tracing modes available, but then we want to go to the editing panel and click on this classify button because we already have those structures traced. You'll see when I hit the classify that on the left-hand side, this nerve bundle was changed to a blue color. That means we've already classified it, but the ones that don't have a classification will be this gray-white color. We can select specific segments to name them. You can select one at a time. You can select multiple by holding your control button. And then in this branch name dropdown, this is where we're going to see those SciCrunch terms. We can navigate to the term that we want to use, that we want to call that specific element. Again, in this case, it will be the inferior cardiac nerve. And as I select inferior cardiac nerve, that selection turns the magenta color saying, yes, this is the inferior cardiac nerve. When I save this file, it's all the points that make up that segment that I've selected will be named inferior cardiac nerve, and they'll receive a persistent and unique identifier that maps back to the ontology. Could do the same thing for whole cells, dendrites, axons, bundles, however you're representing your anatomical structure that's in your sample. One of the questions we get is if you can use multiple organ lists in a single session on a single sample, and the answer is yes. You can go back to that vocabulary services window at any point during your tracing session, choose a different organ list and hit begin, and you'll see all of the list will refresh with the terminology specific to that organ. Importantly, you can also request terms to be added. Say you are interested in naming a structure, but that term doesn't exist yet in the ontologies. We highly recommend requesting terms to be added. You can do that in our software through this link to the Spark Anatomy Working Group's website, where you can submit a Spark term request. The Spark Anatomy Working Group will curate that term and map it and add it to the ontology. And that term will then be available for you once it's been added in the SciCrunch connection in Neurolucida 360 or Tissue Mapper or Vasilucida 360. In the meantime, if you want to name a structure and that name is not available in the SciCrunch list at the moment, you can use a custom name. You can do that by clicking on the color sets icon to bring up the preferences that show all of the different branches or the SciCrunch terms, and add a new branch name. Give it a color if you'd like, and hit OK. And when you do that, that term will be added just to this list at the moment, and you can name that structure with that term. You will not receive a persistent unique identifier at that point because that term isn't yet in SciCrunch. But what Aiden is going to show you is how to update files even more after those terms have been added to the ontologies.
Nice. Thanks for demonstrating that the flexibility of the new branch naming with SciCrunch terms looks very useful. Now I want to show you another tool that has recently been connected to the SciCrunch terminology database. The Sets tool can be found in the 3D window toolbar.
Sorry, Aiden.
Thanks.
There you go.
Right here. And this tool allows users to select many different data elements, such as cell bodies and trees, and group them together as one structure. So you can see here in the video that I am selecting each tree of the yellow cell here and the cell body, which are two different elements in our data files. And I'm going to place them into a group or a set and just give them either a custom name or a name from the SciCrunch terminology list. Now, the Dr. Tompkins lab actually named these cells using a custom name. They put in the electrophysiology IDs to link these cells to their respective electrophysiology data. But you could name cells like these with their cell type, pulling the name from the SciCrunch terminology list. And you can see these lists are exactly like the ones that you saw in the contour selection tool. When you're finished in selecting your name, you can click OK, and your set will be added to the list of sets. And you can see as I select each set, it's going to highlight the individual cells that are grouped under these sets. Now I'm going to show you how to update existing segmentation data. The linked terms workflow allows users to recognize SciCrunch term names and write unique identifiers to the data elements to help make your segmentation fair. And you'll notice in this data file that I have a contour for the stellate ganglion in gray and one in red. And these terms have not yet been linked. And so we're going to update those and link them to a SciCrunch term using that linked terms workflow. So when you select the linked terms workflow, you can see that it's asking for a parcellation scheme that you would like to pull a list from. And I'm going to select the sympathetic nervous system, but I'm also going to select the peripheral nervous system list because for this data file, I have terms in both lists. So you have the flexibility to select multiple lists at once. And when the linked terms workflow loads, you'll see those two parcellation schemes and the terminology lists in separate tabs on the right side of the window here. And then on the left side of the window, you'll see your contours that are unassociated at the top in that top box there. So those are not linked to any SciCrunch terms, and the ones that are associated in the bottom box here. And you'll notice that the gray contour, the cervicothoracic ganglion, is actually associated with a SciCrunch term now, even though it was a custom named contour. And that's because the name of that contour was exactly the same spelling as the cervicothoracic ganglion term within the sympathetic nervous system list. And so the tool automatically associates that term and writes the unique identifier to the data file. Now the stellate ganglion is not associated, but I can easily manually associate that by selecting the stellate ganglion term and the cervicothoracic ganglion term and clicking replace. And that's going to change the name and write that unique identifier to the data file. You'll notice that I have some SOMA terms up at the top here that are still unassociated, and I'm going to leave those as is because that's how the Tompkins lab did it in their data, but you could, for example, again, name those as cell types using the SciCrunch lists. And then you'll see other tabs at the top here for different data elements, such as markers and trees. And so you can employ the same process to update the marker terms and link them to the SciCrunch terminologies. You see most of my markers in this data file have already been associated except for this white one at the top. So I'm just going to search for the junction between the thoracic sympathetic nerve and the cervicothoracic ganglion and select that term from the list and then go ahead and click replace to associate that marker with the SciCrunch term that applies. And now I just want to go back to the contour tab really quickly and tell you about this maintain channel or maintain color option. So you can select this before you associate any of your terms, and it will keep the color of the contour element that you had selected originally instead of what is written to the SciCrunch term. When you're finished, you can just select OK. And you'll notice that this message here is telling me that I didn't associate all of my contours, but we talked about how those SOMAs aren't going to be associated for this data file. So I'm just going to say, all right, I'm ready to proceed. And again, it's saying that some of them were successfully changed, but not all of them because of those SOMAs. But you see now the outer contours of the stellate ganglion have turned pink, indicating that change to the term in the SciCrunch list for the cervicothoracic ganglion has been applied. So I just showed you can link terms and associate elements of your segmentation data with FAIR terminologies for one data file at a time. And now I want to show you how you can apply this process to many different data files at once. We have recently developed the batch process for the linked terms workflow, allowing users to select a folder containing many data files that are similar to each other and associate those elements within the data file with the FAIR terms from the SciCrunch database. This tool helps deal with big backlogs of data from a whole experiment and makes those data files fair and consistent all at once. And you can see on my screen, the process looks exactly like the linked terms, but it's going to show you all of the data elements from every data file you selected to open. Well, that wraps up our demonstrations for the latest FAIR vocabulary tools in MBF Bioscience software. including Neurolucida 360. These tools are soon going to be released to the general public, so keep your eye out for the next NL360 release that has these FAIR tools in them.
Thanks, Aiden. If you're a Spark investigator and would like a license of MBF software courtesy of the NIH, please email us at sparc@mbfbioscience.com to get started. We can provide you with training sessions and demonstrations and help your lab reach their Spark mapping goals. We can also put you in touch with scaffold engineers at the Auckland Bioengineering Institute as you begin or continue the mapping process. In addition, if you have segmentation data from other sources, we'd love to talk to you about converting it to the MBF XML so you can add these FAIR vocabularies and subject metadata.
To make the data output fair and allow tool builders like ABI engineers to ingest the data produced by MBF Bioscience, we have fully documented our file format, and it is available to the public. You can see the link to the file specification on the slide here, www.mbfbioscience.com/file specification. Any tool developers can use this, and if you're interested in creating custom analysis and ingesting the MBF Bioscience data file format, you can use the file specification to do so. You can pull meaningful metadata that we covered today, like subject metadata, and URIs from the segmented anatomies. Additionally, we have a web form link posted on the slide here. So if you have any questions about our file specification, you can go ahead and ask them on the web form. And we have another link at the bottom to our preprint manuscript discussing the useful elements of our data file, why it is important. So go ahead and check that out.
Lastly, we just wanted to show you some things that are coming soon to the Spark portal. We have a segmentation data viewer, so data sets that have been published to the portal that contain MBF segmentation. You'll be able to interact with the segmentation files and check them out in a 3D viewer on the gallery page of the portal, so look out, that should be coming soon. Oh, go ahead, Aiden.
No, you're good.
And we also have MBF has another webinar coming up. We just wanted to let you all know about it, called The Reconstruction of Whole Mouse Brain Volumes from Serial Sections for Registration to the Allen Atlas. I believe our colleague, Dr. Nathan O'Connor, will be presenting that on Tuesday, March 2nd at 11 a.m. Eastern Time. We will, just a reminder, any Spark investigator, if you're on Slack and you have questions for us, you can contact us directly on Slack or on the MapCore-support channel. We're there to help you, and so is the Spark Anatomy Working Group if you have questions about SciCrunch and how those ontologies are created and surfaced on the Spark Anatomy Working Group's SciCrunch page. We're all there to assist.
Thank you guys for attending today and taking the time out of your day. We really appreciate it.